we are going to look into what are the different components of a stirring type bioreactor. A stirring type bioreactor in simple is made up of either glass or stainless steel. The container is made up of glass or stainless steel, or else it could be made up of polypropylene. And the simplest of the stirring type bioreactor is, it is a simple bioreactor. It is a large vessel. It is a large vessel, and in which the cells can be cultured. So, you use it for cell culture. You use it for cell culture, and mechanically it can primarily be stirred. So, that is one of the reasons why it is called a stir type bioreactor, because it can be mechanically churned. It can be mechanically churned due to the presence of propellers. So, it is it is useful primarily in the study of animal culture. Animal culture, though the bioreactors are dealt in botany, predominantly bioreactors are meant for small cell animal cultures. It consists of a stirrer. It consists of a stirrer, and the stirrer can mix the contents. So, a stirrer which is fixed into the sort of a bioreactor will have blades or fans. And the stirrer can propel the sort of a culture. It can propel the sort of a culture, and because it can stir, it provides oxygen. There is more of oxygen which is provided in the sort of a bioreactor, and it is thereby a stirred bioreactor has got lot of advantages when compared to the sort of a flask culture. In a flask culture, you have the nutrient medium which are agitated by movement of a sort of a flask. The movement of a flask is by means of shake culture. So, in a shake culture, the chemical gets rotated, it gets stirred, whereas in the case of a stir type bioreactor, it is more advanced. So, we have here a sort of a delivery system. We have a delivery system, and the delivery system provides the oxygen. And then we have got the sort of a propellers, we have got the churner. So, when oxygen and when churning goes on, there is a formation of foam. And once there is a formation of the foam, the temperature and also the temperature regulates the sort of a foam, and thereby again we have at the bottom we have the sampling port. So, we have a regulated temperature, we have the formation of the foam, and we have a sampling port. So, while taking on the sampling port, we need to exactly take in the sort of a cultures and check it up physically and biochemically periodically. Thereby, we can exactly know what do we have in the sort of a samples. In the samples, we can get the cells, and we can analyze the cells for their sort of a growth. We can know in which sort of a growth phase they are present. We can know the growth phase, whether they are in the exponential growth phase, whether they are in the plateau phase, or whether their cells are in the decline phase, and thereby we can take concern of the sort of an extracted product. We can exactly say the extracted product is exponentially derived and it is economically viable. Now, whenever we take a sort of a bioreactor, we understand bioreactors are large size vessels, they have a precise control of the environment, and there is a sort of a mass culture of cells. There is a mass culture of the cells and of the processed organisms. And when you say processed organisms, it means these are the organisms which have got the sort of a recombinant DNA and its products. So, we have a stir tank. So, a stir tank is nothing but an upright cylindrical vessel. It is an upright cylindrical vessel with a sort of a slant bottom, so that the medium just gets settled at the bottom, and the both ends are closed. Upper end and lower end are both closed, and it is a hemispherical basin. If you understand, it is a hemispherical basin. Now, the reason why it is called a stir type bioreactor is the stirrer is fixed. The stirrer is fixed. And again, there is a mixing of the air and the foam currents. So, there is thorough agitation. So, once there is thorough agitation, it is very easy to enable the sort of fermentation to go on. Now, next we will be looking at typical diagram of a bioreactor. In a very simple diagram, I will be primarily explaining to you the different components of a stirred bioreactor. A stirred bioreactor is meant for continuous culture. We require a continuous culture of the microbial cell or the plant cell which have got the gene of interest. The objective of using a bioreactor is to get large scale of your commercial product. So, every bioreactor has got a pressure gauge. It sets in the pressure gauge because it is just like a sort of a pressure cooker, and it has got a stirrer, and the stirrer is called the mixer. To the mixer, you have attachment of a foam breaker. This is a foam breaker. So, pressure gauge, mixer and foam breaker are on one line. Then to this we have the churning, which is called the sort of an agitator, and the agitator is made up of several lines which are termed as flat bed impeller. The flat bed impeller rotates like a sort of a clock. It rotates like a clock. So, if you take a sort of a bioreactor, it is a stainless steel or a plastic vessel. It has got, if you remember, it has got a hemispherical bottom, and we have several sort of an inlet and outlet ports. Through the inlet ports, we send in compressed air, we get air steam, and we also put in acid and base so that the pH is regulated, and thereby all these are inlet ports. So, through the inlet ports we send in compressed air, steam, etc. The reason is because the component should be sterilized. We are sterilizing at a very high temperature and also under pressure for a given period of time, and then you fill the sort of an agitator with a nutrient broth. The nutrient broth is made up of a defined medium. It has got different essential elements which are essential for the growth of the microbial cell, which has got the sort of a recombinant gene, and we have a nutrient broth, and the entire thing is covered by means of a sort of a cold water pad. There is an inlet for cold water and an outlet for cold water. And here again we have got different sort of a temperature control systems for pH, temperature, oxygen, and they can be regulated by means of different sort of a valves. And thereby in a sort of a bioreactor, when a spent medium or when you have the growth of the cells which is continuously occurring by continuous culture by means of a stirring, the stirring is due to the presence of a sort of a shaft. and the shaft also rotates, and when it rotates you primarily have the mixing or the churning of the medium, and when there is a churning of the medium there is a formation of oxygen, and oxygen bubbles get formed over here, and in the froth there is exactly you can take up the, so from the outlet you can take up the microbial cells and exactly know what is the sort of a how is the method of growth. We can know whether the cells are in the initial stage of growth or in the exponential stage of growth or in the plateau stage or in the decline stage of growth. So, this is in simple a basic diagram of a stirred bioreactor.